Hello and welcome. Today we're diving into the fascinating intersection of synthetic biology and body art, exploring a new frontier in diagnostics using engineered bacteria as living biosensors within dermal tattoos. For years, scientists have been developing wearable diagnostic systems that can give us real-time information about our bodies by measuring biomarkers in our biofluids. Tattoos have emerged as a particularly appealing platform for this. They are small, minimally invasive, can be implanted for long periods, and allow for continuous monitoring directly through the skin, which acts as a diagnostic interface. Tattoos are typically placed in the dermis, a layer of skin between 0.4 and 2.2 millimeters deep. This layer is rich with cells, blood vessels, and nerves, all bathed in what's called interstitial fluid, or ISF. This fluid contains a wealth of physiological information, biomarkers like pH, metabolites, and electrolytes. By placing sensors in the dermis, we can get a continuous reading of these markers, offering a powerful alternative to traditional blood tests. Most existing tattoo-based biosensors rely on small molecules that change color or fluorescence when they encounter a specific target. But what if we could use something more sophisticated? This is where synthetic biology comes in. Instead of simple molecules, we can use living cells, specifically bacteria, as tiny engineered biological devices. Through genetic engineering, we can transform bacteria into micro-machines that can act as highly advanced sensors. Interest in these bacterial sensors is growing rapidly. They can be designed with incredible specificity and sensitivity to detect a huge range of molecules, from toxins and pollutants to critical health biomarkers. One of the key advantages of bacterial sensors is their ability to provide real-time, continuous monitoring. They are living systems, so they can adapt and respond as the environment around them changes. They can even self-replicate and replenish their populations, offering the potential for long-term functionality without frequent maintenance or replacement. But perhaps the most powerful aspect is their programmability. Using the principles of synthetic biology, we can install genetic circuits into these bacteria. This allows them to perform complex computations, like using logic gates to respond only when multiple conditions are met and to execute dynamic response profiles. We can even connect their sensing capabilities directly to a response, such as programming them to produce and secrete a therapeutic molecule on site when a specific disease marker is detected. The research we're examining today aims to demonstrate the feasibility of using these engineered bacteria as living analytics within dermal tattoos. The central idea is to create dermal tattoos containing responsive bacteria-filled microgels. These microgels, produced using a technique called droplet microfluidics, would house engineered bacteria capable of responding to both biophysical cues, like temperature, and biochemical cues, like the presence of a specific molecule. Once produced, these microgels would be tattooed into a skin model. The goal is to show that the embedded bacteria can survive this process and actively respond to stimuli from within the tattooed region. Of course, you can't just tattoo free-floating bacteria into the skin. The researchers recognized the need to enclose the bacteria within protective capsules. This encapsulation is critical for several reasons. First, it enhances the bacteria's functionality, stability, and survival by providing an optimal microenvironment. Second, it helps the bacteria evade the immune system. Third, it ensures precise localization, preventing the bacteria from spreading beyond the tattoo area, which improves detection and minimizes any risk of infection. And finally, especially when working with genetically modified organisms, encapsulation provides a vital layer of biocontainment, addressing important safety concerns. The material chosen for this encapsulation is a hydrogel. Hydrogels are three-dimensional, porous networks of polymers that are highly hydrated, biocompatible, and mimic biological tissues, making them perfect for this kind of application. Within this hydrogel environment, the trapped bacteria can remain active, while small molecules from the surrounding interstitial fluid can freely diffuse through the gel's matrix to be sensed. The specific hydrogel used here is alginate, a material commonly used for bacterial biosensors. Alginate forms a gel in the presence of divalent ions like calcium, creating a stable 3D architecture. Using microfluidic techniques, this gelling process can be controlled to create micro-scale beads, perfectly sized for transdermal delivery via tattooing. Let's now look at how these bacteria-filled microgels were actually made. The researchers used a microfluidic chip made from a common polymer called PDMS. The design of this chip is key. It has a junction where two separate aqueous streams meet an oil stream. This is called a flow-focusing junction. The first aqueous stream contains the engineered E. coli bacteria, a buffer called HEPEs, and a solution of alginate mixed with a compound called Ca-EDTA. EDTA is a chelating agent, which means it tightly binds to metal ions. In this case, it's holding on to calcium ions, keeping them unavailable. The second aqueous stream is similar. It contains the E. coli, HEPEs, and alginate, but this time mixed with a different compound, ZN-EDTA. Here, the chelating agent is holding on to zinc ions. These two aqueous streams are flowed into the microfluidic chip where they meet the oil stream, which contains a surfactant called SPAN-80. At the junction, the force of the oil flow pinches off tiny, uniform droplets of the combined aqueous solution. The surfactant stabilizes these droplets, preventing them from merging as they flow down the channel. Inside each newly formed droplet, a clever chemical reaction takes place. The zinc ions from the ZN-EDTA complex have a stronger affinity for the chelating agent than the calcium ions do. So, the zinc ions displace the calcium ions from the Ca-EDTA complex. This releases a flood of free calcium ions within the droplet. These free calcium ions then immediately begin to cross-link the long alginate polymer chains, causing the entire droplet to solidify or gel into a microgel bead. This ion exchange reaction is performed at a pH of 6.4, which is gentle enough to not harm the bacteria while still allowing for rapid and homogeneous gelation. The resulting microgel beads, now containing encapsulated bacteria, are collected from the chip. When analyzed, these microgels were found to be highly uniform, with a mean diameter of 151 micrometers and a very low polydispersity index of 0.0076. This uniformity is a hallmark of microfluidic production and is important for consistent performance. The concentration of bacteria inside the microgels could also be easily tuned by simply adjusting the amount of bacteria added to the initial precursor solutions. Microscopy images confirm the success of this process. We can see individual microgels where the bacteria, which have been engineered to produce green fluorescent protein, or GFP, emit a localized green signal. This shows that the bacteria are intact and active after encapsulation. The fluorescence is confined within the gels, indicating high encapsulation efficiency and minimal escape of bacteria during production. Now that the researchers had successfully produced these bacteria-filled microgels, the next crucial step was to test their function. Could the encapsulated bacteria still sense and respond to stimuli from their environment? To answer this, they tested two different types of engineered bacteria. The first was designed to respond to a biophysical cue, temperature. These bacteria were equipped with a genetic element known as an RNA thermometer. An RNA thermometer is a segment of RNA, usually placed just before a gene's coding sequence, that acts as a temperature-sensitive switch. At lower, non-permissive temperatures, this RNA folds into a stable hairpin structure. This hairpin physically blocks the ribosome binding site, preventing the ribosome from attaching to the messenger RNA and translating it into a protein. In this case, the gene being controlled was for GFP, so at low temperatures, no green fluorescent protein is made. However, when the temperature rises above a certain threshold, in this case about 35 degrees Celsius, the heat provides enough energy to melt or denature this hairpin structure. The RNA straightens out, exposing the ribosome binding site. The ribosome can now bind and translate the gene, leading to the production of GFP. The researchers placed these thermometer-equipped microgels under a microscope and heated them to different temperatures, monitoring them for 2 hours. At 30 degrees Celsius, there was a minimal increase in GFP expression. The switch was in the off state. A similar result was seen at 35 degrees, which is around the transition temperature. But when heated to 40 degrees Celsius, there was a significant increase in GFP production. The RNA thermometer had switched to the on state, and the bacteria lit up with green fluorescence. This demonstrated that the sensory mechanism worked just as well inside the microgel as it did in a standard lab culture. Next, they investigated a biochemical trigger. For this, they used a different strain of bacteria, this one engineered to respond to a small molecule called IPTG. IPTG is a classic tool in molecular biology used to induce a specific genetic promoter, in this case leading to the production of GFP. The nanoporous structure of the alginate gel, with pores around 5 nanometers, is large enough to allow the small IPTG molecule to freely diffuse in and reach the bacteria inside. When these microgels were exposed to IPTG, they saw a clear dose-dependent response. Higher concentrations of IPTG led to a greater increase in GFP fluorescence. This is a critical feature for any biosensor, as it allows for quantitative measurement, not just a simple on-off signal. Importantly, these experiments were conducted at 30 degrees Celsius, a temperature at which the RNA thermometer from the previous experiment would be off, showcasing the ability to create distinct and independent sensing systems. The researchers also confirmed that these biosensors could respond dynamically. After an initial activation, the bacteria could respond to a new pew by producing even more GFP. Finally, they assessed the stability and viability of the system over time. Over a 12-hour period at room temperature, the bacteria within the microgels were observed to grow into microcolonies, with very limited escape from the gel. Even after 20 hours, these microcolonies remained viable and responded to the addition of IPTG. However, after being left in a buffer for 7 days, the bacteria no longer showed activity, although the microgel structures themselves remained intact. This indicates that the microgels provide a stable containment unit for the lifetime of the bacteria. Having demonstrated that the bacteria-filled microgels could function as robust biosensors, the team moved on to the core objective of the study, tattooing these living analytics into a skin mimic. The skin mimic was made from agarose gel, chosen for its transparency, which simplifies imaging, and its well-established use as a skin-like material with controllable properties. To perform the tattooing, they used the commercial tattoo gun. The microgel solution was loaded into the gun's cartridge. The gun works by using a micro-scale needle array to pierce the agarose mimic, depositing the microgels into the site of the piercing. First, to ensure the microgels could even survive the mechanical stress of being ejected from the tattoo gun, they tattooed them into a simple buffer solution. The gels successfully retained their shape and kept the bacteria encapsulated, confirming their viability for tattooing. Next, they tattooed the gels directly into the agarose skin mimic. To help visualize the gels, they were tagged with a fluorescent dye. After tattooing, they performed what is called a Z-stack imaging, which involves taking images at multiple focal planes to build a 3D reconstruction of the tattooed region. This revealed that the microgels were successfully embedded at a variety of depths, from the surface down to 1.64 millimeters. This is a crucial finding, as it shows the ability to place the sensors within the typical depth range of real tattoos, and to potentially target different layers of the skin.
For comparison, they also tried tattooing unencapsulated, free bacteria. The result was a weak, highly diffuse fluorescent signal spread throughout the tattooed region, with poor positioning. This starkly illustrates the benefits of encapsulation. The microgels protect the bacteria and provide a much more prominent, localized signal. To get an even clearer picture of the tattooing process, they sliced the agarose mimic and imaged it from the side. This revealed the holes created by the tattoo needle, with the tattooed microgels arranged in a neat line within these channels. This confirmed that the tattoo gun was successfully injecting and positioning the microgels. They also noted that the microgels appeared less spherical inside the agarose than they did in buffer, suggesting they are more constrained by the surrounding matrix, again indicating successful embedding. To showcase the versatility of this platform, they loaded the tattoo gun with a solution containing fluorescent dextran, a large sugar molecule that doesn't diffuse easily, allowing it to stay in place for imaging. With this ink, they were able to easily tattoo a variety of user-determined shapes into the skin mimic: a smiley face, a cross, and a star, demonstrating the potential for creating cosmetically appealing and complex sensor designs. The final piece of the puzzle was to confirm that this approach would work in a more realistic skin model. They encapsulated standard tattoo ink into the microgels and successfully tattooed them into a sample of porcine skin, which is structurally very similar to human skin. Images showed that the ink-filled microgels were present in the porcine skin, with the tattoo ink remaining contained within the microgels. This result is a strong indicator that the microgels are indeed appropriate for tattooing into real skin. Now for the culminating experiment. Could the tattooed bacteria sense and respond to a chemical cue from within the skin mimic? To test this, the researchers prepared an agarose skin mimic that was preloaded with the model biomarker, IPTG. They then tattooed the IPTG-responsive, bacteria-filled microgels into this agarose block. The idea was that the IPTG present in the surrounding agarose would diffuse into the tattooed microgels, reach the bacteria, and trigger the production of GFP. After incubating the tattooed block for 4 hours at 30 degrees Celsius, they imaged the sample. The results were clear. In the same locations where the tattooed microgels were visible, a corresponding green fluorescent signal had appeared. This was direct evidence that the bacteria were both contained and protected within the tattooed microgels and were actively producing an interpretable signal from within the tattoos. To see this process in action, they recorded a 2-hour time-lapse video. The video showed that in the skin mimics containing IPTG, the fluorescence intensity from the bacteria steadily increased over time. This response was also concentration-dependent, just as they had observed in the earlier experiments. And crucially, in the control tattoos made in agarose without any IPTG, no response was seen. These experiments definitively confirm that the microgels not only can be easily tattooed, but that they also sense and respond to chemical signals in the exact same dose-dependent manner as they do in bulk solution. This is a very powerful finding, as it means that these bacterial biosensors can be rapidly prototyped and optimized in a standard lab setting, saving significant time and cost, with the confidence that their function will be identical when they are eventually placed into microgels and tattoos. In conclusion, this study establishes a robust and scalable platform for using engineered bacteria as living analytics in a tattoo-based system. The entire process is underpinned by the microfluidic production of bacteria-filled microgels, which allows for the continuous and high-throughput creation of these tattoo precursors. This is a significant advantage over batch production methods and is vital for any future large-scale application. The use of standard, user-friendly tattoo equipment further supports the potential for widespread adoption. The modularity of this system is one of its greatest strengths. The encapsulated bacteria can be thought of as living inks. Different populations of bacteria, each sensing a different stimulus, could be used as different inks. They could be tattooed into the same region or into different regions, enabling the simultaneous sensing of multiple stimuli. The alginate hydrogel itself is also versatile and could be modified, for example, by incorporating enzymes that cause it to degrade over time, leading to a naturally resorbable tattoo. Looking forward, the potential of this platform is immense. The genetic circuitry of the bacteria can be made far more complex. We can incorporate logic gates that require the detection of a combination of analytes to give a descriptive readout, a level of sophistication that is very difficult to achieve with conventional, non-living biosensors. This also opens the door to creating biohybrid tattoos, where these living microgels are combined with non-living electronic sensors, further expanding the scope of detectable stimuli. The applications are broad and impactful. Such a system could be used to monitor personal health through biomarker detection in the interstitial fluid, provide a triggerable response to infections, or even selectively mark regions of the skin for cosmetic purposes or targeted therapies. This work provides a foundational proof of concept, demonstrating a complete pipeline from the high-throughput production of bacteria-filled microgels to their successful deployment as functional biosensors within a tattooed skin mimic. It helps usher in the exciting possibility of wide-scale deployment of synthetic biology within our own skin, creating the next generation of biosensors for both research and societal benefit.